Okay, so today we're going to talk about or start on the macromolecules. One thing that I have, it'll help if I actually let you see what I'm working on here. One thing that I have found to be confusing when we get to this point for most students is breaking down each category and remembering what belongs where. So this first flow chart is more important than maybe you might realize. So I want to make sure that we go through this part first, and then we'll break down each of these macromolecules. So the macromolecules, macro means big, so we're talking about big molecules. Most of these have a monomer and a polymer. Do you guys remember talking about that? How if you've got a chain, a monomer, no, I don't know why that's not working. My lights aren't on. Okay. Okay, so if this is a monomer, a polymer is a chain of the same thing. So a monomer is one, and we're going to talk about monomers and polymers a little bit more. And then the polymer, poly means many. So many of these have a monomer and a polymer. The four major macromolecules are carbohydrates, protein, lipids, and nucleic acid. We are not going to spend time talking about nucleic acid in anatomy. That one's a little more for the college biology class. We spend more time on that, and then biology. But in anatomy, we aren't going to spend a lot of time on DNA and RNA. Just know that those are the nucleic acids. So carbohydrates, we're going to break down the carbohydrates into a couple of different categories. First, you've got complex carbohydrates and simple carbohydrates. They are exactly what they sound like. A simple carbohydrate can be just one sugar, which would be a monomer, or it can be two. So this is one or two sugars. Complex is chains of sugars, or the polymer. And we're going to break down the complex carbohydrates also a little bit more. We're going to revisit this again, so if you don't get it written here, don't sweat it, but realize that you've got complex carbs. There's a few different kinds. There's starch, which is a carbohydrate that is stored in plants. And then there's glycogen, which is a carbohydrate that is stored in the muscle of animals. And then cellulose is a building material. That's the stuff in plants that, let's say when you eat celery, there's always a wad of that stuff you can't chew and you can't digest. Pretty good chance that's cellulose. Cellulose is indigestible to humans. In fact, most animals. The animals that eat grass have a special bacteria living in their stomach that actually breaks down the cellulose for them. They don't actually break it down themselves. So cellulose is a really tough, undigestible, in most cases, undigestible material. All right, then you've got proteins. Proteins, the monomer is amino acids. Then the polymer is just a chain of amino acids, which we call a protein. So you're going to have to know the monomer and the polymer for proteins. You're also, though, going to have to know, that's why this one has stars, because this one's a little different. You're also going to have to know the structural levels. So we'll talk about what those are in a minute. And then lipids. We're going to talk about the three main categories of lipids. There's saturated, unsaturated, and then cholesterol or steroids. Cholesterol is kind of like they call it a precursor for steroids. It means it's kind of the base. It's what you start with, and then you can build different steroids or hormones out of the cholesterol. And then the nucleic acids. So when I look at this first page, this tells me for carbohydrates, I'm going to have to know the difference between complex and simple. I'm also going to have to know the difference between the three complex carbohydrates. Looking at this, I know that protein is going to be made out of amino acids. I'm going to have to know the monomer and the polymer. And I'm also going to have to know the structural levels of protein. For lipids, I'm going to have to know know the difference between saturated, unsaturated, and cholesterol or steroids. And then nucleic acids, really all you have to know is that they're DNA and RNA. So that one's the easy one. All right, now we can move forward. All right, so carbohydrates. Yeah. So we're going to start out with carbohydrates. The one thing that all of these have in common is they are all made out of carbon, hydrogen, and oxygen. And then nucleic acids also contains nitrogen. And because they contain, these all contain carbon, because they all contain carbon, what do we call these macromolecules? Carbon-based. We have another word for carbon-based anything. It's right, it's carbon-based. Starts with an O. Organic. That's the real meaning of organic. We learn organic to mean like, oh, it was made without pesticides and it was grown naturally, free range. I mean, all of these things that we attach organic to. In science, though, in anatomy, organic means that it's carbon-based. Carbon is kind of the main chain. So in all of these, carbohydrates is a long chain of carbons that have hydrogens attached. We'll look at that. Protein. Proteins are a little more complicated, but the main atoms, the main elements you find in proteins are carbon, hydrogen, and oxygen. Same with the fats. Or I was describing fats when I said carbohydrate. Carbohydrate is the glucose. It's strands of glucose. So you've got the carbon ring. You guys have gotten to the point in chemistry where if I drew something like that, have you learned that every corner, anytime you see shapes drawn like that, you can assume that at every corner there's a carbon? Have you learned that in chemistry, I guess is what I'm asking you? Or have you just known that from other classes? Some of you I know already know that. Okay, so if you haven't learned that in chemistry yet, I use that when I draw some of these molecules, that anytime I draw, so a lot of times the shape, this is one of the shapes that you're going to see. I got it right, yes. That means that every corner is a carbon. If you draw it this way, this is another way that you'll see it. Every corner is a carbon. So we're going to draw out these molecules. You're going to see there's a lot of carbon, a lot of hydrogen, a lot of oxygen in all of these. All right. So let's start with carbohydrates. Carbohydrates, they're molecules made of carbon, hydrogen, and oxygen. We call they're sugars and starches mainly. A carbohydrate is a single 5 or 6 carbon ring. We call them fructose, glucose, and sucrose are examples. So what it would look like, let's draw the 5 carbon ring. Looks like a home base plate. And the 6 carbon ring looks like, I don't know, a sign of some sort. A 6 side. So that's how you would draw the carbohydrates. So simple sugars are the monomers for carbohydrates. A simple sugar. So fructose could be a monomer, glucose could be a monomer, sucrose could be a monomer. And they're simple sugars. They're 1, sometimes 2. Disaccharides, di meaning 2, saccharide meaning sugar. 2 sugars, that's still considered a simple sugar. Sucrose is a disaccharide. So they're simple because there are only 1 or 2 of these glucose, fructose, or sucrose molecules. There's only 1 or 2 of the sugar molecules. All right. So I have those 2 outlined in yellow. If you end up taking these notes or printing them, I put those in yellow because these are the simple sugars. Monomer, 5 or 6 carbon ring, fructose, glucose, sucrose. We can also have disaccharides. This is all simple sugars. If we go down a little bit more, here is where we're going to start talking about complex sugars. And that's where I've got the green outline. So complex sugars are going to be the polymers. So I would take a chain of these. Anyway, so now we're getting complex because it's bigger. It's more of them. So now I've got a chain. Here's my monomer, which could be a glucose, and my polymer, which would be a complex carbohydrate. Now, when we have a complex carbohydrate that is made and stored in plants, we call this a starch. So starch is made by plants. Think of a potato. Is that not the starchiest thing that you can, that's like the first thing when I think of any kind of starchy food. First thing that comes to my mind is a potato. Well, a potato is a plant. So if you can remember that starch is found in potatoes, and potato is a plant, you should make that connection between a starch being a complex carbohydrate that is stored, that's made and stored in plants. Then you've got glycogen is the complex carbohydrate that is made and stored in animals. They're both branched. Glycogen is going to be more highly branched, which means that you can actually break it down easier. You can pick off just little pieces of energy as you need it. Which is why we need that, because we're moving more often. So this is the sugar that's in our blood, readily available all the time when we need quick energy. Plants don't ever need to just like quick take off. So theirs doesn't need, they don't ever need to have to like break off just a little bit of quick energy. They can take that little by little, just start breaking down the starch as they need it. they needed more energy for something. Does that make sense? We need it more branched, so we can just start breaking off little pieces of that for quick energy. So an example of that is when you work out. The first, and I don't remember, I haven't talked about it since college biology. The first, is it 3 minutes? The first 3 minutes that you work out, all you are doing is, let's say you're going from couch to running. So you're sitting there in your desks right now, and I tell you to get up and run. The first 3 minutes, is it 3 minutes or is it 30 seconds? I think it's 30 seconds. It goes really fast. Do you remember that? So it's like this very short amount of time, somewhere between 30 seconds and 3 minutes. Very short amount of time, your body reaches into your blood and starts pulling off this glycogen stored in the blood. So that first, I think it's 3 minutes. The first 3 minutes, you're just going on sugar that's in your blood, that's just readily available. And then pretty soon that starts to deplete. And that's when you start to get, like even if you're in really good condition, and I see, some cross-country runners or even people that I know run, at first, sometimes after the first few minutes, even when you're in really good shape, you hit a wall and you start, oh my goodness, I've been running for like months. Why is that happening? Well, you just depleted your glycogen level. Now your body's making a shift into starting to break down some reserves of sugar. So now you're kind of breaking down the ready reserves, and then after 20 minutes, then you start breaking down stored energy. That's why they say you need to work out at least 20 minutes when you do your workouts, because it takes about 20 minutes before your body starts to tap into, on average. Now there are other ways to ignite that process, but in normal circumstances. So we have this glycogen just floating around in our blood, ready to use. That's why that's there. All right, so starch, plants, glycogen, animals. They're all complex carbs. In other words, they're polymers. So here's down here, and then cellulose is just, it's the same as starch, it just alternates the bonds. You can see this, that's just what you call a functional group attached on the outside just to show the arrangement. And that every other arrangement makes it lock into place. Just really locks it into place, and that's why it's difficult to break down, because you can't access, our enzymes cannot access the active site to break it down. If you didn't understand that, you don't have to, and if you did, it was just for further explanation if you do get that. All right, starch, not very complicated, pretty easy to break down. Cellulose, very complicated, or not branched, but arranged differently, does not break down easily. And glycogen then has all these little branches, and it doesn't show, but there would be even more branching because our body is going to be taking little pieces of quick energy all the time. Okay, so now let's just go back one more time. I just want to show you that we have already completed complex carbs, or carbohydrates. They're mainly carbon, hydrogen, oxygen. Monomer is a simple sugar. If you tell me glucose is a monomer, that works. The polymer is a complex carbohydrate. Complex carbohydrates come in the form of starch, glycogen, and cellulose. Starch is a complex carbohydrate stored in plants. Glycogen is a complex carbohydrate stored in animals. Cellulose is the building material used by plants that's indigestible food. Okay. Then we move on to amino acids, or proteins. Oh, I didn't put them in order. So proteins. Proteins are the most varied of macromolecules. They have the most variation. A protein is a polymer made of amino acids. Amino acids is the monomer. Does anybody remember how many amino acids there are? 20. There are 20 amino acids. So out of 20 amino acids, we make all of the proteins that we need. And here's the basic amino acid structure. They're all the same in that they have a central carbon right here. Sorry. Has a central carbon right here that's attached to another carbon plus oxygen. We call that a carboxyl group. Carbon and oxygen, carboxyl. And then an amino group, which you guys, have you learned amine yet? So anytime you see amine means nitrogen. Nitrogen. So eventually you figure that out anytime you see AMIN. So here's our nitrogen, and it fills up with hydrogen. What's different in every protein is what you call the R group. The R group is what makes them different. So you can see here's all of our amino acids. They all have carbon, hydrogen, carbon, nitrogen. They all have that same structure. Here's our amine group, hydrogen, carboxyl. Amine group, hydrogen, carboxyl. That's the same on every amino acid. It's the R group that you can see changes. Some are bigger than others. The simplest is glycine. Glycine just has a hydrogen. Alanine is the next simplest, which has a carbon and 3 hydrogens. And then it starts to get more complicated. But they're all the R group. Now, proteins do all kinds of things in our bodies. Proteins are the workers in our body. So let's say you put your hand down on something hot. Now, the whole process before you actually pull your hand away is that you have sensors in your hand that tell you it's hot, nerves, right? It sends a message up to the brain. The brain says, all right, well, you're going to have to pull your hand away. So it sends the message back down to the hand. All right, you need to pull away. You're getting hot. So throughout all of this, nothing's moved yet. Nothing's going to happen until you have a protein. A protein needs to be there to carry out all of the jobs that need to be done in a body, whether it's a muscle contraction. A muscle contraction is going to at some point need a protein to make that happen. Sometimes it comes in the form of an enzyme, sometimes it comes in the form of a hormone, sometimes it comes in all kinds of different forms. Proteins can come in all kinds of different forms, but they get the work done. Does that make sense? It's so complicated actually that it's hard to say in just one sentence. So let's just say proteins do a lot of work in our bodies. They're the workers in our bodies, in our cells. Okay, here's where we have to learn the structure though. There are 4 levels of protein structure that are going to decide its function. This is why we have to learn this. Because depending on all 4 of these things, that's going to decide what that protein does. That's why we can have 20 amino acids and have like thousands, hundreds of thousands of proteins in our world made out of those 20 amino acids. Because it matters what order those amino acids are in. It matters, once you've got the order, it matters how they hydrogen bond to each other to start to like fold or make a coil. And then once they've folded or made a coil because of hydrogen bonds, then it makes a difference which functional groups are sticking off to the side that might be attracted to each other or repelled from each other. That changes the shape. And then finally, you're going to have different proteins, these complex amino acid complexes that bind together, because a protein isn't made out of just one polymer. Usually it's made out of a few different polymers stuck together. So depending on which polymers stick together. So all of those things play into what that protein is going to do in your body. And that's why we can have so many different proteins out of just 20 amino acids. So here's what we look at. First, the first thing that affects or decides the function is the order, the sequence of the amino acids. So here's our polymer of amino acids. There's our protein. Or it's not quite a protein yet until it's all folded up, but here's our polymer. And let's say this one is alanine, serine, glycine, methionine. You can go through the order, obviously, is going to change what that protein does, right? That makes sense. Depending on which amino acids you put in there and in what order is going to decide the function, what that protein does. I'll come back to that in a second. The next thing, so that's number one, primary protein, is the sequence of amino acids. The second one is, it occurs when the sequence of amino acids are locked by hydrogen bonds. So let's say this one is attracted to that one because of a hydrogen bond. And then this one is attracted to this one because of a hydrogen bond. And then this one is attracted to that one because of a hydrogen bond. You guys remember a hydrogen bond is when a hydrogen with a partially positive charge is attracted to anything with a partially negative charge. Just a background. So it's like a magnetic attraction. It's not a bond that's locking them together. It's more like they're pulled towards each other like magnets. So that's the secondary structure. So it's going to change the shape. Are you going to have this alpha helix, which is more like a spiral? Or are you going to have a pleated sheet? That's all decided by the hydrogens having this magnetic attraction to other parts of this polymer. And they can create different shapes. So the second one is the hydrogen bonds. That's the key. Then the third thing that's going to determine the function is the interaction of side chain molecules. So just for that one, I'm going to quick go back. These R groups are the side chains. So they're sticking off the sides of these polymers. You can imagine looking at how big some of these side chains are, and some of them are small. Well, when we look at this polymer, those side chains are sticking off in every which direction, depending on how they bond. Those are also going to have attractions to each other. So that's what this next, the third part is. The third part is those interactions of the side chain molecules, the R groups. And then the quaternary, the final one, tertiary means 3, quatern means 4, like quarters. The quaternary is when you have more than one amino acid chain sticking together to make a protein. So it'll be like more than one polymer. Here it says more than one amino acid chain or more than one polymer. So up to here, up to the tertiary, everything up here has to do with one chain, all the interactions in just one chain. Then finally the last piece, the fourth part, is putting a few different chains and sticking them together to make the final protein. So those are the levels. Primary is just the sequence of amino acids, the order that they go in, what amino acids go in what order. The second one is the attraction of hydrogen bonds, like that magnetic attraction that gives them either an alpha helix or a pleated sheet. Those really are the only two choices. And then the tertiary is the interactions of the side chains or the R groups and how they interact and pull together or push away from each other is going to change the shape of the protein. And then quaternary when you stick more than one together. All right. Are we here till 20? Okay, good. Because I just have one more thing, because this leads us right into the next thing. Why do we care about the shape of a protein? That's right. If it loses its shape, it loses its function. And the classic example of that is an enzyme. And we had to learn that for biology, for college biology. So for some of you, this should be a review. But enzymes, if you remember, the shape of an enzyme makes a big difference if it works or not. So that's what we were talking about down here. So metabolism, I threw that in there because that's a word that you have to know. I think you guys got that one. I'm not going to spend a lot of time on it. But metabolism is the sum of all the chemical reactions in a cell or in your body. Just all the chemical reactions. So one in particular is enzymes. So here's my enzyme. An enzyme is a protein. So it's represented by this really weird shape, but that shape is not by accident. That shape has to do with the sequence of amino acids, then how the hydrogen bonds pull them, and then how the side groups interact and twist and pull and push away. And then you stick a few of those chains together, and now you've got this shape that's not by accident. And that shape fits exactly with a substrate. Whatever needs to be chemically reacted, which would normally not, this chemical reaction would normally not happen on its own, or it would take so much energy that it probably wouldn't happen on its own. So what happens is the substrate fits perfectly, it binds in. If it doesn't fit, it doesn't work. That's why we care about the shape of a protein. It's so important. Because an enzyme is not going to work if it's not the right shape. So the substrate binds, it causes a chemical reaction. In this case, we're breaking it in half. And then it sends off the products. This whole process can also reverse. It can be the other way. It can be two products come and bind and become one. So this can, depending on, not all enzymes work both ways. I don't want to be confusing that way. I mean, some enzymes are breaking down. Other enzymes are building. So enzymes can either break bonds or build bonds. But that's essentially what an enzyme is doing when it's making a chemical reaction happen. It's either breaking bonds or building bonds. And the enzyme is unchanged. So it's like a tool for making and breaking bonds. I wish that I had, it's kind of like, it's very much like if you had an old root beer bottle. You guys ever drink that old root beer stuff? I love that. But most of them aren't twist-offs. So at the point when you buy it, that cap is one with the bottle until you get the right tool. So you go grab your bottle opener and you wedge it in there just right. If it's not the right shape, it won't wedge in there right. If you don't have a good bottle opener, it doesn't work as well. You got a good bottle opener that wedges in there just right, then you just pull up, and it's a lot easier than if you try to crank on it with your fingers. Now here's the deal. It's possible that maybe if you get your fingernails under there, and you dig and you dig and you dig and you pull, you might eventually get it off. but it's going to take a lot of energy, right? Like, I'm not strong enough. I'm just going to tell you that right now. I am not strong enough to pull that bottle cap off by myself. But all of a sudden I get a different tool, and I wedge it in there just right, and it pops right off with very little energy. It's very much like this. When we are opening a bottle without the bottle opener, it takes that much energy. Probably not going to happen. If I have a bottle opener, all of a sudden it only takes that much energy, and it happens much quicker and much easier. I'm more likely to open more bottles if I have a bottle opener. Same with an enzyme. If you don't have the enzyme there, if the protein is not there, this is how much energy it takes for that chemical reaction to happen. If you have the right tool, if you have the right enzyme, then this is how much energy it takes to make that chemical reaction happen. Whether that's breaking a bond or pulling a bond, making a bond. All right. So that feels overwhelming. So I'm going to stop right there. But I am just going to go back one more time, just like I did with the carbohydrate. Oops. I want to look at protein now. We did all this carbohydrate, complex and simple, starch, glycogen, cellulose. Now we learned about the proteins. The monomer is the amino acid. The polymer is the protein. The structural levels determine its job, its function, because the shape of a protein determines its function. And we will once again have to remember that an enzyme is a protein, and you'll have to remember that picture. Identify what's going on in that picture. An enzyme is lowering the activation level. It's lowering the amount of energy it needs to make that chemical reaction happen by using a tool. And that tool is the enzyme, is the protein. That makes sense? All right. We'll review all of that again before the test. Tomorrow we'll cover lipids and probably break from there for some questions and getting caught up and maybe reviewing anything that's foggy so far. And then we'll have maybe one more day where we cover some of the other odds and ends of chemistry stuff I expect you to know. Most of it's going to be pretty basic. Actually, all of it, I would imagine, is pretty basic after the macromolecules. So macromolecules are a big part, just because that's very much the biology side, very much the anatomy side. How does this affect us and our bodies? All right.